Perfect.
Okay, maybe we can get started. So this is Amanda Sierra, one of the organizers. Rosa, if you can please share the introductory slide.
Yes.
Yes. So welcome everybody. This is the first session of 2025. We have the pleasure of having Michelle Monje today, who will be introduced later. But before, I wanted to remind you who's the team behind that puts all the work behind these talks. At the University of Lausanne, we have Rosa, of course, and Anclés Compagnon, Gloria and Federico, and Giorgio. And in Achucarro, we have Marta Pereira, Fede Sori, and myself. And just to remind you that we are going to have super cool upcoming talks from Gilbert Di Paolo, Arkady Kutorsky, and Stephen Young in the next few months, that you can also follow us on Twitter and Blue Sky, and that you can also check the repository online that we have in Achucarro's website if you can check all the talks because they are recorded in YouTube. So I'm just gonna give the floor to Gloria, who is going to introduce Michelle. Thank you, Gloria.
Yes, so I'm delighted to introduce our speaker for today, Dr. Michelle Monje, Milan Gambier Professor of Pediatric Neuro-Oncology at Stanford University and the Howard Hughes Medical Institute investigator. Dr. Monje completed her MD and PhD in neuroscience at Stanford, followed by a neurology residency at Mass General Brigham and a fellowship in pediatric neuro-oncology. Her research group in Stanford focuses on neuroglia interaction in health and brain in brain cancer, bridging the four neuroscience, immunology, and cancer biology. She has received many prestigious honors, including, just to cite some, MacArthur Fellowship, election to the National Academy of Medicine, and the 2024 Ross Prize in Molecular Medicine. So we are very excited to have Dr. Monje here today, and please, the stage is yours.
Wonderful. Thanks. It's so nice to join you and to get to talk about one of my very favorite cell types. As I think, this audience does not need much of an introduction to, healthy cognitive function depends upon generally intact mechanisms of neural homeostasis and plasticity. And we know that, this does not depend solely on the function of neurons, but really a collaboration between neurons and the various glial cell types, including, very importantly, microglia, which are responsible for modulating the plasticity of synaptic structure and function, as well as regulating neuronal excitability and many, many other homeostatic, developmental, and physiological jobs. Astrocytes, which are important for in their homeostatic state for maintaining healthy synaptic function. And one of my other favorite cell types, the oligodendroglial lineage, which contribute to ongoing myelin plasticity and maintenance of myelin homeostasis. And then in some restricted regions of the nervous system, and in particular in the hippocampus, there's ongoing neuronal plasticity, ongoing generation of new neurons in the dentate gyrus that contribute to some forms of hippocampal memory function. I'd like to focus a little bit on myelin plasticity and homeostasis, because throughout the course of the talk, we're going to discuss the relationship between microglial cell states and its influence on myelin plasticity and homeostasis. Myelin, we now understand over about a decade's worth of work or more, is quite dynamic throughout the lifespan, not only during a protracted period of myelin development, but also throughout adulthood. And this is regulated by the activity of the nervous system. We now understand that neuronal activity and neurons of various forms can induce proliferation of OPCs, the generation of new oligodendrocytes, and establishment of new myelin internodes on previously unmyelinated axons or discontinuously myelinated axons, as well as remodeling of existing myelin. And this we find to be, number one, circuit specific and axon subtype, neuron subtype specific. And there are regionally diverse mechanisms of myelin plasticity throughout the nervous system. This idea of plasticity of myelin encompasses both de novo myelination, the generation of new oligodendrocytes to establish new myelin internodes, as well as remodeling of existing by existing oligodendrocytes of internodes, whereby they become longer, shorter, thicker, or thinner. And these relatively small changes in myelin structure can very profoundly affect the dynamics of a circuit, that the timing of arrival of signals from two places, for example, and this contributes to circuit coordination and, really importantly, to a range of functions. And now in the field, we understand that in the healthy nervous system, this plasticity of myelin is really important for attention, for memory, for various forms of learning. And it makes sense then that when it is deficient, perhaps in diseases, of development, such as neurofibromatosis type 1 neurogenetic syndrome, that this contributes to learning impairments. We also know that one can acquire deficiency in myelin plasticity, and that some of the conditions associated or exposures associated with so-called brain fog syndrome, such as cancer chemotherapy, exposure to COVID-19, both can impair myelin homeostasis and plasticity. And we're going to talk in some detail about that in the next section of the talk. But I wanted to just, before we do, also point out that in diseases characterized by abnormal patterns of activity or abnormal levels of neuronal activity, like epilepsy, there can be maladaptive changes in myelin. And that contribute to maladaptive circuit modifications and disordered circuit function. And we now know that too much myelin can also be a bad thing, and contributes to disease exacerbation and generalized epilepsy, and even reward learning with drugs of abuse, like opiates. But I'd like to focus, given that this is a microglial seminar, on cancer therapy-related cognitive impairment, the dysregulation of myelin plasticity that occurs because of microglial reactivity in this disease context, as well as COVID brain fog. So for the rest of the talk, we're going to talk about neural immune interactions in cancer therapy and in long COVID. And just to give you kind of an outline or preview of the rest of what I'll present, I'll show you data to convince you that reactive microglia in particular states can cause multilineage neural cell dysregulation that contributes to cognitive impairment that follows classical cancer chemotherapy, so-called chemofog, new data showing that cancer immunotherapy can do the same thing, and we'll talk about CAR T cell therapy, and then the same kind of principle of reactive microglial-induced neuron glial interaction disruption also plays out as at least one contributor to the neurological consequences of COVID and other immune challenges. So many of you on this Zoom may know that cancer therapy is related to a persistent syndrome of cognitive impairment called cancer therapy-related cognitive impairment. It's colloquially known as chemofog or chemobrain. This is characterized by impaired attention, concentration, memory, speed of information processing, and multitasking. And it unfortunately affects a large proportion of cancer survivors to a variable extent. There are some chemotherapy medicines that cause this more than others. Different ages of exposure also matter. Being exposed to chemotherapies during brain development is more associated with this syndrome than, for example, in young adulthood. And then the risk of cancer therapy-related cognitive impairment syndromes increases with age from there. In many cases, brain MRIs look pretty normal in people who are having even fairly substantial symptoms, but advanced neuroimaging reveals subtle white matter aberrations and decreased hippocampal volume. And what we've come to learn over the last 25 years or so is that microglia are central to this pathophysiology. All of these different cancer therapies, and I'll show you some examples, cause a persistently reactive state of microglia. This disrupts neurogenesis. It induces a neurotoxic state of astrocytes, and it impairs both myelin homeostasis and plasticity, as well as contributes to altered synaptic pruning. This is something actually that I've been studying for a very long time. So this is a slide that comes from my PhD work over 20 years ago, in which we discovered that reactive microglia prevent hippocampal neurogenesis via direct effects of microglial-derived cytokines on neural precursor cells in the hippocampus. And we studied this in a number of contexts, but one important one was after cranial radiation, which is one cancer therapy that's particularly associated with cognitive impairment. So we find that cranial radiation or another immune challenge like intraperitoneal lipopolysaccharide or some other systemic immune challenge causes reactive microglia in the hippocampus that secrete cytokines, importantly interleukin-6, and interleukin-6 effects on neural stem cells in the hippocampus prevents differentiation, full differentiation into new mature neurons, and this contributes to an aspect of the memory phenotype that in this case the rats exhibit. More recently, now sort of fast forwarding 15 years or so, we began to study how chemotherapies influence cognitive impairment, actually initially hypothesizing that we would find a different kind of pathophysiology, but we found essentially exactly the same thing. What we found was that when exposed to a chemotherapy agent called methotrexate, which is an anti-metabolite particularly associated with cognitive impairment, that there was a direct activation of white matter axon tract microglia. This pattern was really specific. We just saw reactivity in axon tracts, not in gray matter. These reactive microglia in turn induce, and I apologize for the barking dog in the background, induce a state of reactive, neurotoxic reactive astrocytes. And together, these reactive glia dysregulate the oligodendroglial lineage, decreasing the proliferation of oligodendrocyte precursors and impairing the full differentiation of those OPCs. They get kind of stuck in this immature state, which decreases both myelin homeostasis as well as plasticity. And we really do know that microglia are central to this pathophysiology. If we deplete microglia using a small molecule CSF1R inhibitor, this rescues the myelin phenotype and rescues cognition in mice. So at this time, immunotherapies were becoming increasingly promising, increasingly leveraged. And as I'll tell you, I developed a real professional and personal interest in CAR T-cell therapy. And we wondered whether systemic inflammation from cancer immunotherapies could induce a similar kind of glial dysregulation and cognitive impairment. As I think many on this webinar know, CAR T-cell therapy leverages a patient's typically own immune system. So lymphocytes are harvested from the patient. They're then sort of weaponized against a target. So they're given a construct expressing a chimeric antigen receptor. This has an antibody binding domain on one side, and on the other side of this chimeric antigen receptor is activation machinery for lymphocyte activation. So it's sort of one step bypassing many regulatory steps in the lymphocyte. When the cell expressing a CAR encounters antigen, it becomes activated and cytotoxic. And so these are engineered to recognize antigens expressed on cancer cells that are not expressed or lowly expressed on normal tissue. And they've been transformative for hematological malignancies. Difficult to treat leukemias and lymphomas have been cured by these chimeric antigen receptor T-cell therapies. They're incredibly promising, increasingly used. And we're working hard in the field to develop CAR T-cell strategies for solid tumors like brain tumors, and I'll tell you about that. But this therapy is associated with almost every patient has some degree of cytokine release syndrome if there's an immunological response, sometimes very high-grade cytokine release syndrome, and there's a high rate of acute but transient neurological symptoms, sort of a very symptomatic confusional kind of delirium syndrome called immune effector cell associated neurotoxicity syndrome, or ICANS. That said, the long-term effects of this relatively new therapy on cognitive function is understudied. We know that early reports are concerning, that people are reporting, the same kind of symptoms that we see after other kinds of cancer therapies: impairment in memory, impairment in multitasking, impairment in speed of information processing. But long-term neuropsychological testing is just beginning. And so we wondered whether CAR T-cell therapy might cause similar kinds of neuroinflammation, multilineage neural dysregulation, and chronically impair cognition in the way that traditional cancer therapies do. And just to introduce you to the people who've been leading this work, this is work that has been led by Anna Garrity, who's a senior staff scientist in the lab, and she's worked closely with an MD PhD student in the lab, Lehigh Acosta. And I wanted to take a little bit of a detour before I tell you about what Anna and Lehigh found, and tell you that we are particularly interested in CAR T-cell therapy, particularly for a childhood cancer type that my clinical and research life has centered around for many years, called diffuse intrinsic pontine glioma, or diffuse midline glioma, DIPG, DMG. This is a terrible, universally lethal cancer, and we discovered a number of years ago that there was very high expression of an antigen called GD2 on the surface of this type of childhood brain cancer. And so, collaborating with Crystal Makel at Stanford, we've leveraged CAR T-cell therapy to treat first mouse models of DIPG, in which we find that these green and purple tumor cells present in the animals treated with control CAR T cells are eradicated by those that are targeting this particular antigen. So very effective strategy in mice. And we have brought this, this particular CAR T cell strategy to clinical trial that's been ongoing for the last 4 years, for a little over 4 years. And we've had some really encouraging results on this trial. Not every child benefits, but many, many do. And just to show you a couple of examples of this, this was one of our very first patients on the trial, a 21-year-old young man who came to us with this diffusely infiltrative tumor in his brainstem, affecting the strength on one side of his face, sensation, hearing, strength on one side of his body, and gait. And in just a couple of weeks, the CAR T cells shrunk the tumor by about 30%. And with that came really remarkable improvement in his neurological function. He regained motor function on the left side of his face. He regained the ability to open his mouth widely and eat a cheeseburger, which was very important to him, regained hearing, and regained improved motor function on the right side of his body, together with improvement in gait. And I promise to get back to microglia in a moment, but it's really important to understand this therapy and why we're hopeful now that we're moving towards having a chance to worry about the long-term effects of this therapy for these patients. So this is this young man's gait prior to CAR T-cell therapy, the morning of his infusion. And you can see how unsteady he is. He's has a very wide-based stance for balance. There's weakness on the right, which is why he's holding his legs straight, and that weakness is exacerbated as he tries to walk on his tiptoes. He was using a wheelchair for long distances at this point. And as you'll see, he tries here to walk in a narrow line, and he falls and I catch him. And then 2 weeks later, after CAR T-cell therapy, this is his walking in clinic. It's much more stable, much faster, narrow-based. He's able to turn. The right-sided weakness, while present, is improved. And he left this clinic visit and went on a 2-mile hike with his family. Another case of a 30-year-old woman with a spinal cord version of this kind of cancer, very large tumor in her spinal cord, encompassing the entire spinal canal. When we met her, she was wheelchair bound and incontinent of bowel and bladder, also in excruciating neuropathic pain. It's really, really terrible tumor. And over the course, and it wasn't as quick for this patient, but over the course of several months, she had a marked reduction in her tumor, reduced by about 91%. She regained, CSF flow around the spinal cord. Here's the spinal cord. There's a little bit of a sort of smudge of what we call T2 hyperintensity. So we know there's still tumor there, but she regained the ability to walk with a cane. She regained full continence. She was able to come off all of her pain medications because her pain had resolved. So really, really marked improvement that lasted over a year for her before her tumor came back. And then in the best news, we had a child diagnosed at 16 who has had now a complete response. And so when we met him, you can see this very large tumor here, completely obscuring the fourth ventricle, diffuse infiltration of the brainstem, extending back into the cerebellum, also diffuse infiltration of the medulla and of the midbrain. And his tumor kind of melted away very quickly. There's still some signal abnormality here, but you see reemergence of the fourth ventricle, normal CSF space around the pons, nice, normal, dark appearing pons, normalization of the midbrain, normalizing here, medulla continues to look better. This here is the biopsy tract, and it still has some sort of fuzziness around it. But by about between 8 months and a year, really nothing but this evidence of where the biopsy had been taken. And he is now more than 40 months from beginning therapy, no evidence of tumor, major clinical improvements, including needing assistance for walking, to being able to walk independently, return of sensation to his face and body, improvement in hearing. So I am incredibly hopeful right now about this therapy, and it's really kind of a privilege for a universal, for somebody who takes care of kids with a universally fatal cancer to get to worry about the long-term effects of therapy on patients with DIPG and spinal cord DMG. But I am worried about it because I hope that we'll have more patients who are living, years after therapy. So with that in mind and that background, I'm very interested in understanding how CAR T-cell therapy may impact long-term cognition. And we've studied this using a range of different cancer models, and we wanted intentionally to study DIPG, to study this cancer that happens within the nervous system, but also to study, cancers that are entirely outside of the nervous system, to understand how the immune response invoked by CAR T-cell therapy may influence neurobiology, even if it's happening outside of the nervous system. So we studied not only this brain tumor, but also acute lymphoblastic leukemia, a cancer for which CAR T-cell therapy is FDA approved and used very commonly, as well as osteosarcoma, where there are currently clinical trials exploring CAR T-cell strategies for osteosarcoma, but this is a cancer, ALL, acute lymphoblastic leukemia, happens in the blood, but can enter the CNS. So this is a CNS tumor. This is a tumor outside of the CNS that can enter the CNS, and osteosarcoma is a tumor that essentially never goes to the central nervous system, that is exclusively outside of the CNS. And in each of these 3 model systems, we used multiple different controls. Of course, we gave on-target CAR T cells, meaning CAR T cells targeting an antigen expressed by that cancer. But we also gave off-target CAR T cell controls to control for just having these active lymphocytes infused, as well as mock T cell control. So these are cells that have gone through the CAR T cell engineering process, but without expressing a functional construct. And what we find is that in each of these models, the tumor targeting CAR T cell causes cognitive impairment in the novel object recognition test. So this is a test of attention and memory function. Mice are placed first in a chamber with 2 identical objects, and mice are inherently novelty seeking, so they'll explore both objects. We then take them out of the chamber for various lengths of time. I'm showing you the data here for a 5-minute interval. We've also looked at 1 hour and 24-hour intervals. We find the same thing in all of them, where the mouse is then replaced into a chamber with now one of these objects replaced by a novel object. So here's a familiar object and here's a novel object. And a healthy mouse with intact attention, who paid attention to and intact memory and remembered what it had previously seen, will spend a little bit more time with the novel object because, it recalls what that was and it's most interested in what's new. So you see that mice that receive mock CAR T cells or CAR T cells that don't target that mouse's cancer do fine on this test. This is how control mice typically do. They spend about 60% of their time with the novel object as opposed to sort of 50-50 at chance. But mice that have received tumor clearing CAR T cell therapy fail the test and spend equal times with the novel and the familiar object. And this is true in mice that have had CAR T cell therapy for DIPG, for ALL, or for osteosarcoma. But importantly, we use the non-targeting CAR T cell for these cancers as the control, for some of the other cancers. So here, GD2 is the target in DIPG, and when the tumor is cleared by GD2 CAR T cells, we see cognitive impairment. When it's given instead to mice that have ALL, in which there is not GD2 expressed on the cancer, so this is an off-target control, there's no effect on cognition. So there's not an on-target off-tumor effect of the CAR T cells on normal brain causing this. It's the tumor-clearing immune response. And when we examine the CSF cytokine and chemokine profiles after CAR T cell therapy in each of these models, we see prominent elevation of a number of chemokines and cytokines, especially ones like CXCL10, CCL2, CCL11, CCL7, many chemokines elevated in the CSF of mice following CAR T cell therapy for this brain tumor, for leukemia, and for osteosarcoma. And the data that I'm showing you here represents the fold change of control mice with tumors, the fold change relative to control mice with tumors. And we're looking 1 month following CAR T cell therapy. So the initial response is even more robust, but this is the response that persists a month after therapy. Not surprisingly, we find the exact same pattern of axon-tracked white matter microglial reactivity in mice that have received tumor-clearing CAR T cell therapy. So in DIPG, in ALL, and in osteosarcoma, we see this exact same pattern that we had previously seen with chemotherapy of elevation of microglial reactivity, in this case measured immunohistochemically using IBA1 and CD68 co-staining. Now, all of the models that I've been talking about so far have been immunocompromised models. These are models in which we're studying the human tumor and human CAR T cells in an NSG mouse. But of course, we know that in NSG mice, microglia and other myeloid cells are not educated by lymphocytes the way they normally are because the lymphocytes are absent from those models. And so we wondered what happened in an immunocompetent model. And we also wondered, in patients, because they do typically have an intact immune system, when they receive CAR T cell therapy, they're given a standard preparatory regimen of chemotherapy to deplete the other lymphocytes so they don't reject the CAR T cells. And so they're given a regimen of cyclophosphamide and fludarabine chemotherapy. And we wondered what effect those chemotherapies might be having as well. And so we used an immunocompetent model of melanoma, which has been genetically engineered to overexpress the antigen CD19 and CD19 targeting CAR T cells. And what we find is that, again, there is this white matter specific pattern of microglial reactivity. We see this with the chemotherapy alone. And it's even more so, it's even more pronounced when the mice have received chemotherapy and the tumor clearing CAR T cell. We see this both in the in the subcortical white matter as well as in the hippocampal. So what do the microglia look like? We can take a better look at them than just doing immunohistochemistry. So if we isolate microglia and do bulk RNA sequencing from mice, and this particular experiment was done in mice, sorry about that were treated CAR T cell therapy for osteosarcoma, we find that microglia are overexpressing a number of different chemokines. They're in a reactive state, and they're particularly overexpressing chemokines like CCL19, CCL2, 27, 24. This clinical trial has been ongoing for a number of years, and while we do have one child who, has been tumor-free, the majority of children ultimately progress and pass away. And so we have a number of early postmortem autopsy samples from patients so that we can study mechanisms of treatment resistance, and also to look at, how the therapy may have affected the rest of their brain. And so here I'm going to show you human single nucleus RNA sequencing data from 4 patients who were treated on the trial of CAR T-cell therapy for diffuse intrinsic pontine glioma, as well as 3 patients who have the same tumor, but were not on the trial, who donated their brains for research, but were not on the CAR T-cell trial. We took samples, or the pathologist took samples from frontal cortex and subcortical white matter, and then we performed single nucleus sequencing. And what we found was that, number one, all of the microglia from patients who, shown here in blue, who did not have CAR T-cell therapy, all clustered together, and these expressed homeostatic transcriptional state genes. And the microglia from the patients were distinct from the homeostatic cluster. Very few clustered in with the homeostatic cluster. Instead, the patient samples were largely in states of reactivity, and they were somewhat patient specific. So each, human patients, it makes sense, will, who have survived different lengths of time after therapy, who've been, given other therapies in the interim, have different states, but all of the patients who were treated with CAR T-cell therapy expressed had reactive states of microglia. And here you can see just some of the genes we think about a lot in microglia. Here's, for example, P2RY12. You see in gray the control patient samples express high levels of P2RY12 in the patient samples, and essentially each of these other clusters stratified by patient. We see a decrease in P2RY12 and an increase in, for example, SPP1. Looking at the either cytokine-chemokine polarization state or different disease-associated microglial states, we find that each different patient cluster is polarized in a particular way, many towards cytokines like TNF-alpha, IL-1. In one patient, we saw sort of a MIMS phenotype, MIMS and DAM phenotype of microglia. So much more to do, many more patients to study, but it does seem that the human sample here sort of supports the idea that after CAR T-cell therapy, even many months later, that microglia are in a persistently reactive state. So back to mice, the CAR T-cell therapy not only induces microglial reactivity, even if the CAR T-cell reaction is outside of the CNS, it also reduces the number of mature oligodendrocytes in white matter. And this is a consistent pattern that we've seen in other disease states. We see this across each of the different CAR T-cell models. And together with the loss of mature oligodendrocytes, we see a dropout of myelinated axons in the subcortical white matter. Looking in the hippocampus, also kind of as hypothesized, we see that this white matter microglial reactivity in the hippocampus and the hilum of the dentate gyrus is associated with a decrease in hippocampal neurogenesis. And I want to point out that CAR T-cell therapy does not always induce inflammation in the nervous system. It really does depend upon the associated immune response. So we have a second model of osteosarcoma that clears very quickly and efficiently, and at 1 month does not elevate CSF cytokine levels prominently. We don't see an increase in microglial reactivity in white matter. We see intact oligodendroglial cells, intact numbers of new hippocampal neurons, and cognitive behavior is unaffected in this mouse model. So I think it's a really important point that it's not the CAR T-cell therapy per se, but rather the associated immune response that is sort of induced by the CAR T-cell response in most cases. So if we think that microglia are central to this pathophysiology, what happens if we transiently deplete microglia using a CSF1R inhibitor? We find that transient depletion of microglia using a CSF1R inhibitor decreases subcortical white matter microglia by about 80%. This partially rescues the number of oligodendrocytes, and it rescues cognition on the novel object recognition test. We've done a variety now of microglial depletion strategies. We find that this rescues cognitive function in all of the other cancer models as well, and that we can allow the microglia to repopulate after this transient 2-week depletion, and that doesn't alter the cellular or behavioral rescue as long as the inflammatory stimulus is gone. So after the tumor clears, then if microglia are depleted, when they come back in a homeostatic state. So what is emerging is the concept of immunotherapy-related cognitive impairment, in which anti-tumor CAR T-cell therapy can induce an immune response within the CNS characterized by elevated CSF cytokines, chemokines, and white matter microglial reactivity. There's consequent dysregulation of oligodendroglial homeostasis and hippocampal neurogenesis that contributes to this impairment in attention and memory. And if we transiently deplete microglia, allowing them to repopulate using CSF1R inhibitor blockade, alternatively in other experiments, we've also instead used chemokine receptor blockade. Both of these strategies mitigates these effects in the subcortical white matter and rescues cognition. So in the last part of the talk, I want to highlight some, striking clinical parallels between chemofog and COVID, or the cognitive brain fog that occurs associated with COVID. I have to share that in the very beginning of the pandemic, already knowing the results of many of these experiments, I was very worried that we were going to see in COVID sort of cognitive syndrome that looks a lot like chemotherapy-associated cognitive impairment, because we know how profoundly immunogenic COVID appeared to be. And clearly, just even a couple of months into the pandemic, people were flooding neurologist offices complaining of this exact same sort of cognitive syndrome that we see after cancer therapies. And so I wanted to study this in my lab, but was in the midst of lockdown. And so reached out to a wonderful immunologist named Akiko Iwasaki at Yale, and we've been working together on this. She had developed a mouse model of COVID-related brain fog. And she had developed a mouse model of mild COVID, and we studied it from the perspective of brain fog. So in patients, just like after cancer therapy, in many cases, standard neuroimaging is unremarkable, but more advanced imaging techniques reveal subtle white matter aberrations and decreased volume of both limbic and, in this case, olfactory structures. This is a problem that affects a large proportion of COVID survivors. One early meta-analysis, chiefly looking at people who had been infected with the ancestral strain of COVID, found that 1 in 4 people experience persistent cognitive dysfunction in at least one domain. There are about 400 million people worldwide who, as of 2024, were estimated to be suffering from long COVID, the larger syndrome of long-term effects, which frequently involves cognitive symptoms. So we wondered whether similar glial dysregulation might contribute to the cognitive symptoms after COVID. And as I mentioned, I reached out to Akiko Iwasaki, who's an incredible collaborator, and many, many people from my lab and from her lab have been working together to address these questions. She had developed a really useful mouse model of mild respiratory SARS-CoV-2 infection. So in this mouse model, mice can only be infected in the respiratory system. Only the respiratory system expresses human ACE2 and is infectable by SARS-CoV-2. These mice don't get very sick. They don't lose any weight. They continue grooming well. They run around their cages with normal levels of activity. There is no infection in the brain. And yet, when we look at their nervous system, they have markedly elevated levels of CSF cytokines and chemokines, not just 1 week after infection, but persistently through 7 weeks after infection. And just as predicted, we see this very specific pattern of white matter selective microglial reactivity after SARS-CoV, this mild respiratory restricted SARS-CoV-2 infection in multiple strains of mice, both CD1 mice and BALB/c mice. Looking together with Avinath and others at a cohort of humans, of people who died in New York City in the spring of 2020 with relatively mild COVID. They died for other reasons or died suddenly and were found to have COVID infection at the time of autopsy. When we look at their their cortex and their subcortical white matter, we also see this pattern of enrichment for microglial reactivity in the subcortical white matter. So turning back to the mouse model, we did single cell RNA sequencing of microglia from these mice, and we found some really interesting changes. Number one, there was a chemokine expressing subpopulation of microglia. It's small, but fairly prominent. And this really evokes what we found in the CAR T cell treated patients of upregulated chemokine expression by microglia. When we look at this larger population of homeostatic microglia, what's interesting is that while they are technically, expressing a homeostatic transcriptional pattern, their expression of homeostatic microglial genes are altered with decreased expression of, genes like SAL3 and TREM2 and upregulated expression of genes like ApoE. So they're homeostatic, but they're not quite homeostatic. And looking more closely at how these post-COVID microglia, this is looking a month after COVID exposure, how they, how their transcriptomic states compare to other described disease-associated microglial states, we see a lot of overlap, but also some unique features of this post-COVID microglial state. As predicted, with this white matter specific microglial reactivity, we see a loss of oligodendrocytes in the subcortical white matter and in the hippocampal white matter, together with a dropout in myelinated axons. And we see this after this mild COVID exposure at 7 days after infection, as well as persistent through 7 weeks after infection. Now, comparing the degree of myelinated axon loss after COVID and after methotrexate chemotherapy, we see that these are fairly similar. Also, as predicted, we find that there is a dropout in the generation of newborn neurons in the hippocampus, both at 7 days and 7 weeks. Now, we noticed that in the CSF of the mouse model, there was elevated CCL11 chemokine levels. And this was really interesting to us. And actually, they go up over time. So while they're elevated at 7 days, they're even more elevated at 7 weeks. This was interesting because some really beautiful work by Tony Weiss-Carré's lab and Saul Villeda showed a number of years ago that CCL11 can influence hippocampal neurogenesis and alone can contribute to cognitive impairment. So we wondered whether this was an important chemokine in this context. And when we took a look at, together with David Petrino, at a cohort of patients being treated in New York City for long COVID, those who self-reported brain fog symptoms, shown here in red, had a higher overall level of CCL11. Now, there's quite a wide range of CCL11 levels. There's quite a bit of variability in this data set. And so we wondered, if there were other factors that might be associated with the higher levels. And so we did a multivariate analysis and found that those individuals who had a history of autoimmune disease who had brain fog syndrome and a history of autoimmune disease had higher CCL11 levels. So again, kind of indicating that it is the immune response to the immune stimulus more than the immune stimulus itself that is responsible for the pathophysiology. So then we wondered if CCL11 was sufficient to induce some of this pathophysiology. So we took healthy mice and we administered CCL11 intravenously. And what we found, fascinatingly, was that was sufficient to induce microglial reactivity only in the hippocampus. We didn't see cortical or subcortical white matter microglial reactivity with CCL11 administration, We just saw it in the dentate gyrus, kind of highlighting how different populations of microglia in different anatomical regions, or perhaps in different neural circuits, are differentially regulated by the same kind of factor. Together with the elevation in microglial reactivity in the hippocampus, we saw that CCL11 administration was sufficient to decrease hippocampal neurogenesis, consistent with what the Weiss-Correa lab had previously shown. And so taking a step back, long COVID is a complex disease, and there are, many potential contributors to the neurological symptoms, which are not mutually exclusive. But one major aspect of this appears to be this consistent, immune challenge, in this case, respiratory COVID, inducing reactivity of microglia and elevation of CNS cytokines and chemokines, consequently decreased oligodendrocytes, dropout of myelinated axons, and reduced hippocampal neurogenesis, all of which contributes to the neural circuit dysfunction and cognitive impairment. In addition to this, which we see playing out after cancer therapies as well, additional mechanisms can contribute to cognitive impairment after COVID. Importantly, autoimmunity, autoantibodies, for example, neuroinvasion of SARS-CoV-2. It's very controversial how often that happens. It certainly can happen. Reactivation of other infections, so reactivation of latent herpes viruses, in particular EBV, seems to be a contributor in many patients. Microvascular disease, including thrombotic events, also contribute to the pathophysiology. And some beautiful work from Katerina Eglasu's lab recently showed that this small vessel damage results in fibrin leakage, which induces a cascade of events that contributes very importantly to the pathophysiology that's seen. And certainly in patients who have had more severe COVID with multi-organ failure, hypoxia, metabolic disturbances, all of that can negatively affect brain and brain function. We wondered in the last couple minutes, I promise to finish up soon, whether other respiratory immune challenges might induce similar pathophysiology, if there was something really special about COVID. And what we found was that mild respiratory H1N1 influenza causes the same kind of pathophysiology, but not as persistently. So we found transient subcortical white matter microglial reactivity and oligodendrocyte loss. This was present at 7 days after flu in young adult mice, but not at 7 weeks. In contrast to the transient nature of the subcortical pathophysiology, in the hippocampus, we saw much more persistent pathophysiology. Microglia remain reactive, and there's a loss of neurogenesis even at 7 weeks. And thinking about, what's similar and different about in influenza and COVID, we see both shared and distinct CSF cytokine chemokine changes. For example, CCL11 is elevated throughout in both diseases, and perhaps that helps us understand this persistent and more prominent hippocampal pathophysiology. TNF-alpha and IL-6 decrease after mild COVID, but increase in the context of flu. And in contrast, CCL2 and GM-CSF, CXCL2 remain elevated after COVID, but resolve after influenza. So much to learn about the role of each of these chemokines and cytokines. But what is becoming clear is sort of an emerging principle in brain fog syndromes, in which various cancer therapies, particularly myeloid activating therapies, as well as other immune challenges, cause a persistent state of inflammation and prominently of microglial reactivity that, through mechanisms that need to be better understood, dysregulate neurons and glial cells, and in particular affects myelin homeostasis and plasticity. And that this contributes, we think, importantly to neural circuit dysfunction in a potentially reversible way and cognitive impairment. So immune challenge related cognitive impairment, kind of thinking about all of these different disease states together. In conclusion, we find that multiple non-CNS immune challenges can induce an immune response in the CNS characterized by elevated CSF cytokines, chemokines, and white matter microglial reactivity. Consequent dysregulation of oligodendroglial homeostasis and myelin contribute, we think, importantly to the impaired attention and memory. And I think fascinatingly, different chemokines cause distinct patterns of microglial reactivity, which suggests that various immune challenges, which will elicit different cytokine chemokine profiles, may increase risk for overlapping yet distinct constellations of neurological and psychiatric symptoms. Targeting this microglial reactivity or targeting multi-chemokine signaling mitigates these neurobiological effects. Lots and lots of people to thank. I want to highlight, especially Anna Garrity, who did a lot of the work that I showed you, as well as Anthony Fernandez Castaneda and Lehigh Costa Alvarez, collaborators Akiko Iwasaki and Pei Wen Liu, funding sources, and thanks so much for your attention. Hopefully we have some time now for discussion and questions.
Thank you, Michelle. Really was an amazing talk. And now it will be the turn of Anne-Claire, who will be handling the questions.
Yes, you hear me well? Yeah?
Yep.
Perfect. So yes, if you want to ask a question, you can ask in the chat directly and I can read it for you. And otherwise, you can just raise your hand and I will ask the question. And I will give you the possibility to ask yourself. So we have a few questions in the chat already. So from Dr. Oleg Butovsky. So you have actually several questions. So he said, Michelle, wonderful work on CAR T-cell microglia crosstalk. A few questions. Have you explored whether microglial activation can be assessed using TSPO imaging in patients treated with CAR T-cells? If so, have you investigated how this translates to the molecular phenotype of microglia, such as the induction of STP1, as you demonstrated? And this approach could serve as a valuable tool for understanding the pharmacokinetics of CAR T-cell microglia interaction. Maybe I can ask the other one after you reply for this one.
Okay. So, hi, Oleg. Nice to see a question from you. We have not, I have shown you now everything we have done so far. It's hot off the press, just got the single cell sequencing recently. So this is kind of where we are with the human samples. So we have not, but that is a really interesting suggestion. And do I know how the CAR T cells impact microglia that are cytotoxic to the oligodendrocytes? Excellent question. I suspect it has to do with astrocytes. And we're looking at this now, whether microglial induced neurotoxic astrocyte reactivity then causes this relatively rapid dropout in oligodendrocytes. We see the loss of oligodendrocytes within days. And so I think that's what is probably what we're going to find, and we certainly see states of reactive astrocytes in the patient samples, but we haven't yet, done all of the appropriate necessity and sufficiency testing in mice to say that we, know that that's the link between it. But it's what I hypothesize is the important thing.
And then you had a third question. Did you find evidence of a direct interaction between CAR T cells and microglia in situ?
In mouse brain tissue, the CAR T cells are really localized to the tumor, and in the tumor, they are definitely interacting with microglia in the tumor, but distantly in the cortex and subcortical white matter, we really don't see that. At least we haven't captured it. And in some work that I haven't, I just, for the interest of time and focus, didn't show, we can recapitulate the subcortical white matter microglial reactivity with just intravenous administration of other chemokines. And so while CCL11 might be affecting more the hippocampus and less the subcortical white matter when administered IV, CCL2, for example, affects the subcortical. So I do think we have a building story about kind of a chemokine relay, in which peripheral chemokines are through probably brain borders, then stimulating, white matter microglia to then begin to express particular chemokines that I think are perpetuating the pathophysiology in the CNS, but still kind of working out a lot of those details. Thank you for the questions.
Thanks, Michelle. Thank you.
Thanks. So we have another question from Evangelia Kyriakidou. Amazing talk. I would like to ask how these CAR T cells are cleared of the brain. Was the blood-brain barrier disrupted in patients with mild COVID or the mice, so peripheral immune cell could enter the brain?
So, we know that even without blood-brain barrier disruption, there's quite a bit of immune cell trafficking between the CNS and the periphery, and work by Yoni Kipnis and Beth Stevens, and many others is showing us how this trafficking occurs. in CAR T cell treatment of mice, we can label the CAR T cells and kind of watch their trafficking into the CNS. And if we give them intravenously, about 7 days after intravenous administration, they appear in the meningeal vasculature. And then we see them start to infiltrate from there into the place where the tumor is, in this case, the brain stem, clear the tumor, and then we think they exit through the meningeal lymphatics and hang out in the cervical lymph nodes. So they're trafficking just as lymphocytes, I think, normally traffic. There doesn't need to be blood-brain barrier disruption for them to get where they're going, which is interesting.
Okay, we have another question.
Sorry.
Okay, we have another question from James Ross. Which treatment-induced microglia population do you think is responsible for the decreased cognitive performance in mice? Or do you think other cellular populations or events, like the CAR T cells themselves, are responsible for the decrease in performance?
I think that it is the subcortical white matter microglial reactivity that is induced by the CAR T cell associated immune response that is responsible for the cognitive impairment.
Okay. And next question from Brian Campos Salazar. Amazing talk, Dr. Monje. This cognitive impairment after CAR T cell therapy, as you showed, is rescued using the CSF1R inhibitor. Would the initial activation of microglia be a TLR4 mediated response?
It's a good question. I simply don't know what's triggering the reactivity of the microglia, particularly when the stimulus is outside of the nervous system. Presumably it is a relay. One possibility is that it relates to cellular trafficking from the periphery into the CNS. And another, particularly because we can recapitulate much of this now with just chemokine administration, relates, we think, to chemokine signaling.
Okay. Then Rosa, if you want to ask your question yourself.
Me too.
Yes. Thank you, Michelle. Beautiful talk. I have a question regarding the experiment that you discussed with the depletion of microglia or the chemokine receptor blockade with the CAR T cells, where you showed that basically you have a mitigation of the side effects. And I was wondering, has this an impact on the tumor regression, for example?
I'm so glad you asked that. It does not, it turns out. So we looked, we can monitor the tumors using, we engineer them to express something called luciferase, then we can do in vivo bioluminescent imaging of the tumor burden. And in many of the experiments, we depleted microglia after the tumors had cleared, but we did a set of experiments where we cleared we did microglial depletion during tumor clearance, and they cleared just fine. So in this case, the microglial depletion doesn't affect tumor clearance. In separate work, separate unpublished work, we have looked at, we're looking hard at the role of myeloid cells, microglia, and peripheral, myeloid cells that are recruited to the tumor in hindering CAR T cell efficacy. They're suppressive. And we find that there's a specific window in brain tumors where depletion of microglia actually augments CAR T cell therapy, but it's not the entire time. So early in the CAR T cell response, for brain tumors in particular, it seems like you may need the macrophages around. So the timing really matters, but in the experiments that we did, we still got full tumor treatment efficacy with myeloid depletion, and much of that was done with the leukemia model. It didn't affect CAR T cell efficacy.
Thank you. Thank you.
So if maybe I can ask a last question. It was an amazing talk, Michelle. And I think it was very inspiring that you took this journey from the basic science. I remember that Science 2003 paper. I've cited your paper like a million times, all the way to your involvement in those clinical trials. I think that's super inspiring. So I was wondering if you would have like some sort of advice for young people in the audience thinking how to do this movement. How can they really go from this basic science to the translational science? How did you do it in your career?
I thank you for asking that. I follow, number one, if you follow the science, it usually takes you in a good place. So I didn't intend to continue studying microglia, but I just kept coming back to them because they kept being so important. So, I think following the science is important. And I think following what you love is important. So, doing the things that you find compelling, the questions that really move you, will bring you in the direction that you need to go. As a clinician scientist, it was natural for me to, think about what was really happening in my patients as I saw them have, challenges and difficulties with therapy, and then to come back to the lab and try to answer those questions.
That's great. Thank you. So I think we can probably shut down the series for today. Thank you, everybody. It's right 6 o'clock. Perfect timing for everybody. So just remember that we will be back in a month and that you can find the recorded talk in the website. We will also share the link tomorrow via Twitter and Blue Sky. So thank you, everybody. Thank you, Michelle. And everybody, if you want to say goodbye, just turn on your camera, say thank you to Michelle, and thank you to everybody. Good night.
Thank you, Michelle.
Here.
Thank you.
Bye, everyone.
Bye.